Welcome students. The topic for this video lecture is vacuoles. Students, vacuoles are non-cytoplasmic areas present inside the cytoplasm. And if we see their structure, they are surrounded by a single membrane called tonoplast. So it is very important point students that the membrane which bound this structure that is called tonoplast. Next point student, these are formed by expansion and pinching of endoplasmic reticulum. So endoplasmic reticulum has a role in the formation of these vacuoles. Next point, these are small in animal cells, but these are quite large in fungal cells and plant cells. So it is very important to note down that in plant cells they are small and maybe many number, but in fungal and plant cells they are generally large in size and less in number. Next student, depending upon the contents that what does they contain and functions, they are of four types. Number one, Cell-Vacuoles. Number two, food vacuoles. Number three, contractile vacuoles. This is contractile vacuole. And number four, air or gas vacuoles. So let us discuss them one by one, that what are these four types. Let's start students with sap vacuoles. So sap vacuoles are student fluid filled vacuoles which are separated from the plasma membrane with the help of a membrane already discussed, that is tonoplast. Next student, number of sap vacuoles in case of animal cells and young plant cells is very less. But in case of mature plant cells, what happens? These small vacuoles fuse together to form large central vacuole which occupies up to 90% of the volume of the cell. Next student, the fluid that is called sap, it contains many salts, sugars, amino acids, esters, proteins based products and water soluble pigments such as anthocyanins. So what we have discussed, that sap vacuoles have fluid that is called sap, and in animal cells as well as in younger plant cells their number is many, but in case of mature plant cells they are fused together, that is small vacuoles, and forming large central vacuole which occupies more than 90% of the volume of the cell. Next students, in plants the tonoplast membrane helps in transport of number of ions and other materials against concentration gradient into the vacuoles. Student, what does it means? That concentration of ions and material is very high inside the vacuoles as compared to cytoplasm. So when more number of ions and materials are entering into the vacuoles, they are moving from low concentration to higher concentration. So when things move from low concentration to high concentration, this is called against concentration gradient. And when things move from high to low, then that is called along the concentration gradient. So it is very important to note down that why I'm saying here against concentration gradient. Next students, cell sap causes osmotic uptake of the water. So it means that when water has to enter into the vacuoles, it is entering by osmosis, because it is important for the water relation of the plants and maintains of cell turgidity. If cell has to remain hard, solid, then it is very important, then it should have enough amount of water inside its vacuoles. So how the water enter into the vacuoles? With the help of a process that is called osmosis. Next student, let's discuss the second type of vacuole, that is food vacuoles. As the name indicates, they contain food inside them and they are mainly present in the cells of protozoans such as amoeba, paramecium, protists, several lower animals and phagocytes of higher animals. And food vacuole, how it is formed? It is formed by fusion of phagosome and lysosome. So basically food vacuole has food inside it and it contains many digestive enzymes also inside it, with the help of which that food is digested. So what are food vacuoles? First of all, they are present in lower group animals as well as in phagocytes of higher animals. Second, they are formed by fusion of phagosome and a lysosome. So if they are fused by containing lysosome, so it means they have hydrolytic enzymes, and that hydrolytic enzymes help in digestion of the various types of nutrients that they contain. Next student, contractile vacuoles. Contractile vacuoles are present in some protistans, some algal cells, especially students which are found in water. And contractile vacuoles are mainly responsible for osmoregulation and excretion. Excretion is, student, the removal of excess waste from the body, and osmoregulation is maintenance of ions and valence of water inside the body. So contractile vacuoles are just like our kidney, because our kidney is also performing the same function, that is osmoregulation and excretion. Last type, student, that is air vacuoles and gas vacuoles. These are found in blue-green algae, purple and green photosynthetic bacteria, and as the name indicates, they store many metabolic gases. They regulate buoyancy, means in case of aquatic bacteria, they also help them to float in the water, and also they protect against harmful radiations. So in this way, student, we have completed all the four types of vacuoles. First type was, that is sab vacuoles. Second, contractile vacuoles. Third, we have discussed about food vacuoles, and lastly about gas vacuoles, which contain simple gases, regulate buoyancy and protect against harmful radiations. Now let's start with the function, student. So first function is that vacuoles maintain osmotic pressure and turgidity, and also helps in osmoregulation. So three functions are included in the same point. At first, they maintain osmotic pressure. So if osmotic pressure will remain, then turgidity will remain there. And second, in case of contractile vacuole, they also helps in osmoregulation. Next function, student. Vacuoles provide buoyancy, mechanical strength and protection in plants. Third function, student, transport of various types of ions and materials is supported by tonoplast against the concentration gradient. And last function, student, they also responsible for storage of food and waste materials. So in this way, students, we have completed all the four functions. So with this, our this video lecture is completed. In our next video lecture, we will start with another cell organelle. Till then, take care. Thanks.